Yeah, so in the last lecture, Dr. Getz mentioned that they are trying, using the mutation analysis, you can try to find genes that are recurrently mutated. So in general, the hypothesis is that if the tumor is recurrently mutated or the genes or pathways recurrently mutated, they are likely drivers, right? And in this include tumor suppressor or oncogene. So tumor suppressor prevents tumor from happening, whereas oncogene, they drive the tumor growth. And very often when you have a tumor that's recurrently mutated, it could be also gain of function mutation or loss of function mutation. For example, very often in the tumors, what you see is a gain of function mutation on the oncogenes, which means that this gene normally can drive cell growth, but it needs some external signal. Usually it's a growth hormone receptor. It needs to have the signal to turn on. But with a mutation, now it can just keep growing without outside hormone signals. And that's a gain of function mutation. A loss of function mutation instead is if this gene normally is expressed to suppress the cancer from uncontrollable growth, and suddenly this gene is lost, then the cell can keep growing. And so from the mutation, how can you see whether something is either a gain of function mutation or loss of function mutation, right? You could look at some mutation patterns, including the copy number variation patterns. You could also look at their gene expression patterns. Of course, Gary Gass mentioned last lecture that these are all hypothesized. You still need functional experiments to really figure out how this really happened. But of course, some genes can be both tumor suppressors and oncogenes, which are more complicated depending on in which cancer type and exactly where that mutation happens on the gene. It could have opposite effect potentially. And so we talked about in the last lecture this Gistic Score, which basically look at how often a particular, this is looking at copy number change, right? How often this type of copy number change happens, and also the amplitude. Is it 3 copies or 5 copies, or you lose a copy? And then using this, you can look at whether this is a significant difference. So the idea is if something is repeatedly amplified in many tumors, not only they occur frequently, when they occur like 3 times, 5 times, or 10 times, that's usually a very strong indication this is an oncogene. Because you amplify it, then the patient will get tumor, right? So the copy number give you information. If you have a copy number loss, then the patient have tumor. That's more likely a tumor suppressor. You can also look at, we mentioned in the last lecture on the specific mutations, and very interesting talk on Tuesday, that mutation rate is not even across the genome. Different locations have different mutation rate. This is potentially related to epigenetics. So basically, if it's open chromatin, they are less likely. So if this region has open chromatin, the genes are highly expressed, they are less likely to be mutated, because very often the gene needs to be expressed. If you have a mutation, it's less likely that's good for the tumor. It's like if you have a car, you randomly take things out. How often is it running better? Most of the time, it will not run very well. Just occasionally, you have a mutation that will make the cancer cell grow much faster. And so most of those mutations will be eliminated because of selection. At the same time, in regions where there are gene expression happening, there are also good DNA damage repair mechanism. They will detect there's something going on. So there is transcription coupled DNA repair mechanism. So highly expressed genes if there are any mutations, they're also likely to be repaired. So it's only in those these regions that are epigenetically silent, not many genes expressed, and like no good repair system, then they get mutated a lot more. And so with this, you can figure out whether mutation happen a lot or not. And when you look at this profile, which we also saw on Tuesday, when interesting observation is that if this is a cancer type that appears in children's, they are less likely to have a lot of mutations. But if it happens in older people, naturally you'll have more mutations. So for example, there are patients who take cosmetic surgery to take out some, like the bags on their eyes. And when you take this out to look at their mutations, if it's coming from older individuals, you will see a lot of mutations, even though they don't have cancer, they just have more mutations, because over time, we accumulate a lot more mutations. And also the tumors that face outside, these are tumors that, skin or lung, because we breathe, and also the GI system, those actually face outside. They face the foods, so they deal with outside stimulus all the time. They have overall higher mutations. Whereas the tumor that face inside, such as brain or blood, you can imagine those really, and also say prostate, they don't really face outside. They usually have fewer mutations. And you can look at the mutation pattern very often to see whether this is an oncogene or not. For example, for MYC, if you look at where the mutation happens on this gene, it's kind of scattered. But most of the time, if you look at many, many different cancer types, most of the time the mutation happens as a copy number amplification. So that just the same gene you just amplify this region, they can have many, many copies. And if you look at the expression, MYC is highly expressed in many, many cancer types. That's usually an indication this is an oncogene, right? Whereas in another case, this is also interesting. You don't see so many copy number gain. But if you look at the mutations, most of these locations don't have mutation, but this one location at G12 locations, there are like 1,300 mutations happening in this exact same location. That's a gain, it's a very clear gain of function mutation. I guess this is where the KRAS needs interaction to be activated, but with the mutation, they don't need the signal. They can always constitutively be on. And so this is clearly a gain of function mutation on oncogene. So this one is kind of more complex. If you look at this mutation, it's kind of all over the place. And also you see a lot of these missense mutation. Green is missense mutation. Missense mutation means that it's an amino acid change. There are also many truncating mutations from the beginning to the end. You can see here, from the beginning to the end, truncating mutations is that you're gonna lose this gene. But also you see more blue, these are deletions. So this is actually a tumor suppressor. You either lose it from copy number or from a particular mutation that basically the rest of the gene are no longer expressed. You have an early subcodon, and so you lose the expression of this gene. That's a clear indication of a tumor suppressor. There are some more complicated patterns. For example, this one, you see P53, it's a very well-known cancer gene. In all the cancers, you don't see a lot of cancer copy number change, but if you look at the mutations, there seem to be some early, you see truncation, a lot of truncations early on, but then there are also missense mutations that happen 400 times. This gene, there's some hypothesis that the wild type gene is a tumor suppressor. So if you completely lose it, the tumor has problem with the DNA damage repair. But interestingly, if it's mutated just in those locations, then it can potentially have oncogenic effect. It can actually promote the tumor growth. Not necessarily a complete gene loss, right? So you can see that's getting a bit complicated. This is another complicated pattern as EZH2. You can see in different cancer types, sometimes it's a point mutation, sometimes red is amplification, and sometimes it's a deletion, and this is complicated. If you look at the expression, it seems to be overexpressed in many cancers, so maybe it's an oncogene, but it could also potentially be a tumor suppressor gene. So you can see it could be quite complicated. If you look at all the tumor mutations from a pan-cancer, all the cancer type together, the blue genes are all the very well-known cancer driver genes, whether it's an oncogene or tumor suppressor. The red ones are kind of novel, but they have clear cancer association, and there are also some really novel ones. The biggest surprise from all these mutation analysis with TCGA or expression analysis is that there are a lot of mutations on chromatin regulators. And because of this, a lot of drugs are now developed to study chromatin regulator changes. So we'll talk very briefly about chromatin regulator mutation as well. Questions?
I have a question. So has anyone combined the GWAS study as well as ATAC-seq study to look at sort of like SNPs and changes, the SNPs that confer the epigenomic changes and thus, and thereby confer to like inherited variant to the like population level somatic mutation, sort of things like that.
So by the way, in the last module, we talk about a disease, right? The first lecture, the guest lecture was talking about more germline mutations.
Yeah.
These are, maybe the mom and dad are both okay, and suddenly the child has a spontaneous mutation, or maybe this is just a SNP that happened in 5% of the population, right? That's germline. All of your cells carry the same thing. Here, we're talking about somatic mutations. It doesn't happen in all the cells in your body. It only happens in that tumor compared to your adjacent normal or blood. They are quite different things, usually. I would say germline mutations, if it's a very rare disease, it could be a very strong effect. You have this mutation, you get the drug. But if it's kind of appearing in 5% of the population, it's probably not a very strong effect. Somatic mutation, it depends on, you can see how often they appeared in the tumor. It's probably the more frequent, the stronger the effect. Yes.
And also, I have a question on the one of the factors that has different expression level in different type of cancer cells. So for that one, because like I suspect this data is from the TCGA, which is like the bulk RNA data. So does anyone like look into like each of these cancer data and do the single cell to see whether there's different expressions because of the specific cell type, and or also because like the different cancer related tumor microenvironment that kind of drives the
everybody is interested in doing single cell. Unfortunately, the TCGA tumors are already frozen. It is not in single cell suspension, so you cannot do single cell RNA-seq on the original TCGA tumor anymore. But but scientists are getting the fresh tumors, getting the single cell suspension. Now they can do either SMART-seq or 10x genomics single cell sequencing. We have a little section at the very end. I'll show you. Okay. So
sorry, also one question.
Yeah.
I just wonder, like, what is the logic behind like a mutation happening many times on one site is a signature of gain of function mutation? And conversely, why, like, if we have like many mutations with low count,
like this, right? Yeah. So
is a sign of like loss of function.
Yeah. So for example, in this situation, normally this gene has its, say in this case, a normal function in order to say interact with other protein, or it needs a growth signal to grow. But then suddenly you have this one mutation, this gene can be constitutively on. That's recurrent. You really need this position to be on for everything else to happen. It doesn't happen if the mutation happens in another location of this gene. And so when you have very, very focal mutations, very often it's a gain of function mutation. It's a key place, because if this position is a loss of function mutation, then it doesn't matter have to be this space, right? You can just lose it early on. You can have a truncated mutation, you lose the whole gene. That should also work. Right. And so this type of mutation, the IRB, is like, you can see you have copy number loss, the whole gene is gone, or you have this truncating mutation. There's an early mutation. The rest of this gene is not even there. Is not expressed. Right. So when it's really scattered like this, it's usually a sign of loss of function because it doesn't matter where it appears. Every mutation makes the gene work less well. It could be completely gone as well. That's usually a sign that it's a loss of function mutation. Right. It's again, you can imagine if you have a car, you take out parts and you're trying to see when does it make the car run better. It has to a very specific part up. You take that thing, suddenly the car runs better. It's very rare, but most of the time you take parts off, the car doesn't work so well, and that's what you see in this RB case.
Yeah.
Right.
That makes sense.
Okay. All right.